morning, everyone, again. Thank you for joining us at our webinar From Complexity to Clarity, Understanding the 2025 EU GC by GC Guideline for MOSH and MOAH analysis. My name is Roberto Ronzoni, and I will be your host for today's session. I'm the manager of Macro Analytical Division. I have a chemistry degree, and I've been working in Neotron since 1998. These are our speakers. Barbara Moretti, she is the head of packaging department, the Neutron lab where MOSH and MOAH are analyzed. She has a chemistry degree, and she's been working in Neutron since 1999. Next up is Cristiana Venturelli. She's been working in Neutron since 2007, and she is a senior technician analyst. Finally, the floor will be for Erika Vellati. She has a degree in chemistry. She has been working in Neutron since 2007, and she is a senior GC analytical specialist with a focus on GC by GC technique. This is our agenda. We will start with a brief corporate and Neutron presentation. Then we will dive into the topic, firstly with Barbara's speech, an introduction to MOSH and MOAH, latest legislative updates, and new GC by GC guideline. Then Cristiana will show us the main instrumental technique involved in analytical control of mineral oil and their connection to risk assessment. Finally, Erika will share with us the potentiality of GC by GC technique. At the end of our webinar, we'll have a Q&A section. We receive many questions during the registration, and we selected the most frequent ones. But if you have any question during the presentation, feel free to use the Q&A section or chat box, and we will address those questions at the end of our program. Let's start with a brief introduction about our group. Cotecna is a Swiss-based company headquartered in Geneva with offices and labs across the globe. It's a leading provider of testing, inspection, and certification services. Today, we now have over 8,100 employees with more than 180 offices and laboratories. Our trusted network of professional and certified laboratories provide expertise across our 5 key sectors, namely agriculture, food safety, metals and minerals, consumer products, and certification. Neotron is the biggest laboratory of Cotecna Group, located in Modena, in the north of Italy, where more than 550 employees work in 10,000 square meters of laboratory. By managing more than 1,000 samples per day and more than 5,000 accredited tests. These are the field of testing activity which Neutron has activated during the 50 years of its history in relation to market and customer requests. So before starting, we propose you a poll question. Please take a moment, 1-2 minutes to give the answer to this question that will appear on the screen. So the question is, how significant do you think the impact of European regulation and guideline on mineral oil will be on your business in the near future? Very significant, minimal, or negligible? Please, your opinion is important for us. Okay, there are the results, more than 100 responses. So the topic is very hot, so very significant or significant are the main choices to this question. So let's start to this topic that is interesting for you. I leave the floor to Barbara presentation.
Thank you, Roberto, for the kind presentation, and good morning to everyone. Now, let's start with the first question. What are MOSH and MOAH? Mineral oil hydrocarbons include a different group of chemical compounds derived mainly from crude oil, but also produced synthetically from coal, natural gas, and biomass. And they are characterized by several mixtures of hydrocarbons, containing thousands of chemical compounds of different structure and size. Mineral oil are essentially composed of two different chemically structured types of fraction. The primary fraction is made up of MOSH, Mineral Oil Saturated Hydrocarbons, that consist of a complex mixture of saturated hydrocarbons with open chain or naphthenic structure. Within these elements, MOSH analogous must also be considered. These substances group are particularly relevant, like polyoligomeric saturated hydrocarbon, POSH, saturated oligomers formed in the production of plastic material like polyethylene and polypropylene, which make up the main component of most plastic packaging. Polyalpha olefin, that are synthetic polymers processed to various lubricant or hot melt adhesive, and mineral oil refined product include all other mineral oil refined, approved, and used as additive and processing aid, such as paraffin waxes. Due to their chemical similarity, MOSH analogous are very difficult to separate from the MOSH fraction using conventional method, which can result in higher MOSH content. The secondary fraction is composed of the MOAH, Mineral Oil Aromatic Hydrocarbons, a mixture of highlight alkylate aromatic with at least one aromatic ring system. These molecule properties increase their chemical reactivity. In fact, MOAH fraction brings a higher concern relative to the MOSH fraction. Due to the large number of alkylated compounds present, typically thousands of compounds that cannot be separated chromatographically into individual components, the mineral oil chromatogram appears as an unresolved complex mixture. In general, MOSH-MOAH ratio is 4:1, and it is a common tool that can be used to indicate a mineral oil contamination. This is our history from the past to the future, with more than 14 years of experience in mineral oil analysis. At the moment, we have 4 HPLC GC FID equipments and 1 GC by GC TOF-MS FID instrument. Mineral oil contamination has been talked about for many years, but the issue is set to take a hot topic for a long time in food safety. As can be seen from the notification posted on the European Commission RAF's window, since the beginning of the year, the border controls are continuing and several matrices are involved in control, like tropical oil, cereal, vegetables, and dietetic foods. It is increasing the importance to identify the source of contamination in order to remove it, and it's important to understand that all the entire food chain is involved. Mineral oil can further be present in food through several sources: use of lubricants for machinery, processing aids, food and feed additives, migration from food contact material, and through environmental and transport contamination. Understanding the source of MOSH and MOAH contamination is essential for developing effective prevention strategies. Today, the complexity of the issue is well known, and addressing the contamination pathway is becoming more and more challenging. As you can see, all this kind of contamination could have a typical chromatogram profile that generally take the form of the hump. And the peculiarity of mineral oil analysis is that on the basis of distinct chromatograms, we can hypothesize the nature of the contamination. On the basis to the last EFSA opinion of 2023, MOSH fraction is not of human health concern, but bioaccumulation is possible. So a margin of safety exposure must be considered, and in case of high MOSH value, mitigation measures have to be provided. In the MOAH risk assessment, two different evaluations have to be undertaken. One for 1 and 2 aromatic ring part, for which insufficient toxicological data are available, and one for the 3 up to 7 aromatic ring for their genotoxic and carcinogenic nature. In the last year, the European Commission as working on a regulatory follow-up to the EFSA opinion. In fact, a draft regulation of maximum limit for MOAH is ready. On March 11th, the European Commission notified to the World Trade Organization of 2 proposed regulations on mineral oil hydrocarbons and 1 proposed recommendation. The 3 measures concern, first, a proposal for a regulation aimed to introducing a regulation 915 on maximum level of certain contaminants in food, limits of mineral oil aromatic hydrocarbons, MOAH, in various food products, including herbals, dried spices, and food supplements. These limits will enter in force the 1st January 2027. A proposal to amend the regulation 333, the regulation concerning sampling and analysis method for the control of trace elements and process-related contaminants in foodstuff, to include sampling and performance analysis method for the official control of the MOAH level in food. Finally, proposal for a recommendation for the monitoring by member state and food business operator of the MOSH and MOAH level in food during the 2026-2029 period. In this proposed regulation, the MOAH maximum limit are defined on the basis to the fatty food content. 3 different categories are defined. Product with a fat content lower than 4% with the limit 0.50 mg per kilo. Product with a fat content between 4 to 50% with the limit 1 mg per kilo. Product with a fat content higher than 50% with the limit 2 mg per kilo. Some exception and different implementation timeline for specific cases will be listed in the annex. For example, the limits to be set for food supplement, spices, and dry herbals will be set at 10 mg per kilo from the 1st January 2027 to the end of December 2029. After this period, the limit will decrease to 5 mg per kilo. One other exception is for tea and herbal infusions. The maximum level will be applied to instant tea, instant herbal infusions, and tea and herbal infusions that are used as ingredient in the food. Together with the limits for MOAH, an update to the regulation will be also published to amend the regulation 333, that is the regulation concerning sampling and analysis method for the control of trace elements and process-related contaminant in food, to include sampling and performance analytical method for the official control of MOAH levels in food. Ensuring reliability, accuracy, and robustness of analytical data is fundamental. For this reason, the urgency of fully validated analytical method must be highlighted. In the draft version of this regulation, some criteria will be fixed, such as recovery and limit of quantification for MOSH and MOAH fraction. For example, it is necessary to highlight that the limit of quantification for food supplements, spices, and dried herbal is 5 mg per kilo. One other important point is referred to the specificity. It is introduced the necessity to use a comprehensive chromatography, GC by GC, when is needed, for example, in presence of interferences. Regarding GC by GC, the European Commission published guideline in the last November 2025. The main objective of this guidance is to promote the harmonization of GC by GC analysis, chromatogram interpretation, and reporting result for qualitative characterization of MOSH and MOAH. This guideline is focused in qualitative interpretation of mineral oil fraction using GC by GC as a complementary technique to HPLC GC FID that remain a reference method for the total quantification of MOSH and MOAH. The guideline is divided in three parts. First, GC by GC analysis. Second, interpretation. Finally, reporting results. This guideline offers a clear criteria for the standardized interpretation of the data and comparable report across different laboratories. One other important tool reported in GC by GC guideline is the flow chart for the decision during the mineral oil analysis. If the MOAH results are lower than the specific limit or MOSH are lower than indicative level, the HPLC GC FID analysis it is enough and we can stop and no more other tests are required. In case when a sample analysis by LC GC FID exceed a limit by taking consideration of the expected analytical uncertainty or the presence of interferences is suspected, the confirmatory GC by GC is required. One other important point defined in this guideline is when a sample exceeds the indicative level, the sample can be declared not in compliance only if the GC by GC characterization confirms the presence of mineral oil markers. But now it's time to go in deep in analytical procedure, and I leave the floor to Cristiana Venturelli. Thank you for your attention.
Good morning, everyone. Thank you, Roberto and Barbara, and thank you to the audience for joining us at this webinar. My name is Cristiana Venturelli, and today we will explore the analytical techniques and the risk assessment evaluation of mineral oil contamination. Firstly, let's focus on the generic method now in use for the quantification of mineral oil. MOSH-MOAH analysis has been specifically designed for a sensitive and rapid detection of mineral oil contamination in accordance with the JRC Guideline 2023, and it consists of a sequence of procedures that lead to the final quantification of the contamination. Naturally, each food matrix follows its own specific pathway, but in general the approach can be summarized as follows. After homogenization, the sample is dissolved in hexane and saponified to remove the lipidic part, enrich the sample, and improve the analytical sensitivity. Subsequently, a liquid-liquid extraction with hexane is performed in order to remove some matrix components. After that, there is one of the most critical steps of the process, that is to say the epoxidation, essential step involved in the removal of MOAH interferences, like olefins, natural polyunsaturated compounds, monoterpenes like carotenoids, squalene, and serine, as you can see from the image, that coagulate during the HPLC separation and show white peaks in the chromatogram that can lead to an overestimation of MOAH fraction. MOSH fraction present natural interferences too. In this case, another cleanup technique becomes necessary. This step consists in the use of an aluminum oxide column for the removal of biologic odd numbered and alkanes and natural paraffins. The determination of true fraction is obtained by the use of HPLC GC FID online couple system that performs a complete separation and quantification of MOSH and MOAH fraction. As said, this is a generic approach, but in the huge variety of food matrices can generate a deviation to this principal pathway, leading to a case-by-case approach. For the mineral oil analysis, there are only two standardized analytical methods specifically used for two different matrices, infant formula and vegetable oils and fats. Hereinafter, you can see the main step of the two methods for which Neotron actively participated to the final drafting. Our laboratory is accredited for matrices of standardized methods, and above oil and infra formula, the list is expanding continuously, comprehending lecithins, cereals, oilseed, paper, and cardboard. Besides the standardized and accredited methods, another tool to help laboratories to reach the robustness of the method is the participation in proficiency testing. Neotron participated to proficiency testing organized by Eurolab DTU Institute, and it often participates to various PT for different matrices in circuits like DRRR, DGF, COI, and Proof HES. Due to its complexity and along with MOSH and MOAH analysis, a risk assessment approach could be strategic. So let's focus on the main steps of the risk evaluation. First, risk identification that can comprehend the classification of all potential sources of contamination across processing environments, for example, equipment lubricants, raw material, packaging material, for example, recycled paper and plastic, considering factors such as fat content, processing methods, and storage condition. The second step is the quantification of contamination. In this step, methods for quantification, LC GC FID must be taken into account, and when the limits of this technique are too effective, other steps are needed. So, ZC by ZC technique can support the analysis of complex mixtures, giving back both qualitative and quantitative data. Finally, it must be highlighted the importance of risk mitigation strategies. For this matter, two main points has to be considered. 1, reduction, management, and elimination of the risk. 2, risk acceptance, when the cost of the mitigation risk is higher than the cost of the presence of the risk. But keep in mind that the risk acceptance can be taken into consideration only if the contamination is below the limits. So, considering all the points discussed, it is clear that the collaboration with stakeholders would be recommended in order to share information and best practices for managing MOAH contamination by working with the supplier, regulatory agency, and industry groups. But let's look together at the following real case to better understand the risk assessment approach. Here you can see an example for MOAH contamination in vitamin soft gel capsules. This case study clearly shows a MOSH contamination at a relatively high level. Based on the preview consideration regarding the importance of mitigation strategies, in this case, an a posteriori risk assessment approach was proposed to the client. Therefore, starting from the finished product, we decided to break it down into its raw materials and perform more analysis in order to identify the source of the contamination and at the same time characterize its nature. Considering the similarities in the chromatograms and the presence of 2 amps, we can conclude that the capsules were contaminated by both paraffins and oil mixture. Most of the sample analyzed are not this directly to evaluated, and the complexity of the matrices, together with the limitation of the analytical technique, considering both extraction steps and the lack of specificity of the FID detector, inevitably leads to high challenges. So I'm going to show you the results that can be evaluated in the case of complex result in order to decide whether to proceed with further investigation using ZC by ZC technique. For example, you can see the chromatograms of an olive oil that show low values of both MOSH and MOAH fraction and chromatographic profile without significant interferences. In this case, it is not considered necessary to proceed with further investigation. Differently, in this second example, the chromatograms referred to a tropical oil reveal that the MOSH autumn is optimal, while the MOAH fraction show interferences and a positive result that may be overestimated. In this case, bidimensional ZC by ZC analysis is recommended only for the MOAH fraction. In the last example, we see chromatograms of a vitamin mixture that both show very high values clearly due to the presence of interferences. What can we do in this case? Here a possible way to handle it. First, work on optimizing the extraction method using a matrix-based approach. As mentioned before, the high variability of food matrices must be considered and managed on a case-by-case basis. Then proceed with a qualitative confirmation of the actual presence of contamination in order to assess any potential overestimation of the quantitative data caused by interferences. However, the true goal is to combine confirmation and quantification of the hypothetical contamination into a single step-by-step using GC by GC TOF-MS FID technique. But for a deeper discussion of this complex topic, I will now leave the floor to Erika Vellati.
So before to pass to go to the Erika's speech, we ask you another moment of your participation with another poll question that is okay on the screen and now. So you have a few minutes to answer to this question that is how often do you have samples analyzed using GC by GC? Okay, in this case the results, the response are more distributed on the 4 choice that we leave. So you know that GC by GC it is a complex technique, quite recent in the MOSH MOAH determination. And to have more information about this technique, I leave the floor to Erika Vellati for her speech about GC by GC technique.
Okay, welcome. I hope you can see my screen now. Thanks, Roberto, for the floor. Thanks to all of you for joining me. My name is Erika Vellati, and today I'll talk to you about the application of GC by GC, so a comprehensive gas chromatography technique in MOSH MOAH analysis. In particular, I will try to answer for you to some tricky questions, starting from why we need a comprehensive analysis like GC by GC, and when it should be applied, how to configure GC by GC instrumentation according to the European guidance of November 2025, what is possible to see in a GC by GC plot, and what extra info can be obtained with respect to the traditional analysis that we have seen before, and how we should report what we have found. Even though the guidance is strictly about qualification, I'll add some few words about quantification in GC by GC, and then I'll jump to some summary and conclusions. So why we need GC by GC? Well, because mineral oil contamination is really hard to detect. We have seen that they have the same analytical behavior of vegetable food grade oils, except the presence of few markers. And searching for mineral oil traces at the end of a complex food chain or a complex analysis sounds like trying to solve a puzzle without knowing the original picture and maybe with pieces missing or not belonging, yes, to the original one. In addition, contamination can happen in every step of food processes. Many levels of contamination can be summed up in the final product, and we must deal with extreme variability in mineral oil composition, so crude, refined, synthetic, heterogeneous, all mixed up, and extreme variability also in samples to be analyzed. Well-standardized procedures are applicable only to vegetable oils, and for other matrices like food supplement, herbal, spices can still be very challenging. In addition, contamination can come also from packaging and can sum up to other contamination still present. From a strictly analytical point of view, we must say that the traditional procedure seen before of sample pretreatments plus LC-GC FID analysis works generally well, but has some limitation. For traditional vegetable oil samples, generally occurs from 10 to 15% of cases, but this percentage tends to rise with challenging matrices, food supplement, herbals, and so on. So starting from the sample preparation, sometimes it's really hard to find a balance between applying purifications or not. Purification tends to change the original sample composition and so the mineral oil composition, and can lead to a loss of MOAH, especially from 3 to higher number of rings, and the destruction of some mineral oil markers. for example, dibenzothiophenes. But on the other way, without them, GC per GC columns can be easily overloaded, and the further optimization process should be applied with different purifications, procedures, and strategies. We must be aware that HAMP is a chromatographic trace. That means that your compounds are not well separated. So maybe different classes of compounds of interest or not are hidden inside. Performing a valley-to-valley integration doesn't mean that you are cutting away interferences, because all these peaks start and then to the baseline. So we must take in account that we are doing an approximation and most probably overestimating the result. In addition, we can be sure that all these peaks are interferences, because FID is not a specific detector. Finally, LC separation efficiency in defining MOSH and MOAH fractions, so setting the boundaries between them, can vary in time and with particular sample composition. This means that you can have MOSH and MOAH colluting in both fractions without knowing it by the use of FID detector only. And internal standard mix check can be totally effective in confirming MOSH and MOAH fractioning, because, of course, it's a standard mixture and can behave different from analytes that are trapped in the food matrix. Many of these limitations can be easily detected and overcome by GC per GC analysis. So let's start with an example. We have seen this plot before. This is an oily vitamins premix for food supplement. When looking at this plot, we see that we have this strange hump because it's too narrow. We suspected some interferences above and suspected also a MOSH contamination because the MOSH output was very high. When going to the GC by GC analysis, we see that the hump is totally out of the MOAH classification space. Some terpenic interferences are still present, so are confirmed, and after investigation of the spectra of the both fractions, mineral oil was even confirmed. Another example, in here we have this spicy cereal snack with a total fat content around 25%. So this will give us a maximum level of 1.0 milligram per kilo. Analyzing the plot, we see the presence of two amps. Most probably the second one is the MOAH one, but we have still interferences on it. And performing the traditional valley-to-valley integration, we quantified about 1.5 milligram per kilo of total amount of MOAH. So this will be not compliant. Going to the GC per GC analysis, we found out that the real MOAH hump was the number 1, not the number 2, that was made of terpenic and olefins interferences all over the plot. And quantifying only the first hump, we found out that it was 0.72 milligram per kilo, so a compliance for this sample. Well, this is the flowchart decision. We have seen it before, and it talks to us when we just need to do this analysis. I summarize all the examples in here when we need it. Well, every time we have an output that exceeds maximum or indicative levels, leading us to a compliance. Every time we don't have a clear view of the hump, we have strange shapes, we suspect some MOSH eluting. Every time we have interferences, we are not sure that there are real interferences. Every time the ratio of the MOSH and MOAH is not connected to a typical mineral oil. Or we are just looking at, contamination sources, packaging, lubricants, or we are testing some new analytics procedures, and we want to see if our pretreatments are good or not. Well, how to configure GC by GC? The starter toolbox to do this analysis is made of course, an LC silica column to do the fractioning between MOSH and MOAH, and the reverse column configuration. What does this mean? Well, in GC by GC, you have two columns, the primary and the secondary, and the phase of the first and the second give us the configuration. In reverse column configuration, we have a mid-polar plus non-polar one. So in between between these two columns, we have a modulator that can be cryogenic or flow. Cryogenic is better. And you need, of course, an injector capable of doing large volume injection. SV, so solvent vapor exit, retention gap can be optional. Of course, at the end, you need a detector and a TOF MS or an MS able to give you some identification. If you are lucky, just like us in Neutron, you also have FID for quantification. But what we see in a reverse column configuration, let's see how MOSH and MOAH are in the space. Well, in first dimension, so on this, compounds elute in increasing polarity and from more to less volatile. In second dimension, compounds elute from bottom to top according to decreasing polarity. So this gives us the MOSH elution space that starts from the top to the mid, and the MOAH elution space from the mid to the bottom. What's in between, of course, all the compounds with intermediate polarity of both fractions. MOAH elutes just a little slightly to the right because they are more retained in first dimension. What you need, starting from the basic, to do your framing of your space, of course, you need a mix of alkane from C10 to C50, your internal standards that you've used before in a traditional analysis, and a pH mix for the bottom. And of course, you need a mineral oil reference. What you use in Neutron is a MOMIX that will be purchasable from LICO. For MOSH fraction, we can identify two macro classes of paraffins, yellow, and naphthalene, the orange. And for the MOAH, we can classify by number of rings, starting from the pHs in the bottom and by degree of alkylation. Because with increasing alkylation, polarity decreases, and we see that those lines tend to go to the top of our chromatogram. Also, heteroatomic aromatic rings with sulfur can be detected, and we will see that are very important MOAH markers. Let's see in details all these classes, starting from the MOSH. Well, we see the linear alkanes elutes in a line with peaks equally distributed by C number increasing. Their distribution between odd and even give us a clue on the nature of the oil. No prevalence of odd or even means most probably a mineral oil presence. If you are in an odd prevalence case, most probably we are dealing with a natural vegetable oil and allox purification should be applied. Or in the opposite case, with a prevalence of even carbon number, we must probably have some packaging contamination. Branch isoalkanes, they elute between the line of a normal alkanes, and with alkylation increasing, they tend to move to the upper part of the chromatogram. In particular, pristane and phytane are very important multi-branch alkanes because they come from the fossilization of chlorophyll and so are MOSH markers. Regarding naphthenes, they generally tend to elute under the paraffin line, and depending on the structure, they can be linear or cyclic. They can, yes, be in different parts of the chromatogram. Styrenes and opanes are polycyclic and are important MOSH markers, while decalines, alkylated decalines, and perhydro PAHs tend to be detected when we are in presence of an hydrogenated oil. Of course, when we talk about MOSH coelution, we just need to deal with synthetic lubricants or packaging contamination. And so be prepared to a very long list of acronyms, starting from the PAO, polyalpha olefins, that are synthetic isoparaffins, generally in the range of C8 to C12 carbon atoms that are used as lubricants and can be easily detected by the presence of these clusters that are slightly above the alkyl line. Then we proceed with the PO, polyolefin oligomeric hydrocarbons that come from plastic packaging, and depending on the type of plastic, polypropylene, polyethylene, and on the density of the polymer used, we can see that the POSH, that are fully saturated, but we can also have POM, monosaturated, cyclic, open change, and so on. When we are in presence of multi-layer food packaging, most probably we will see the typical ROCH pattern. What are these ROCH? They are resin oligomeric saturated hydrocarbons, and that comes from hot melt adhesives. And I've seen before, when talking about MOSH and MOAH separation by LC, sometimes mostly non-polar terpenes and interferences with low polarity can fall in the MOSH fraction instead of the MOAH one. For the MOAH fraction, as we have seen before, we can classify them by number of aromatic ring and degree of alkylation. In first dimension, we have the increasing of the number of aromatic rings. In second dimension, we have, of course, the more alkylation, because when we sum up all of this info, yes, we see that we can draw these lines, these classes by number of rings and degree of alkylation, slowly bending to the top by effect of alkylation. By crossing the PAH's pattern with the C number elution lines, is it possible to predict the degree of alkylation and number of aromatic rings without doing any data processing, just looking at the chromatogram. Every time a ring of 5 or 6 carbon atom is saturated, a plus 0.5 is added to the total number of rings. And we have seen that unfortunately, MOAH with aromatic rings larger than 3 can be unfortunately destroyed by epoxidation. A particular class of circulated rings, which are diisopropyl naphthalines, need a particular mention because their presence gives us a clue about inks contamination, and probably from recycled paper board. When refining processes of the oil, such as hydrogenation, are applied to oils, also more fraction undergo this hydrogenation, with the result of a mixture of aromatic and saturated rings, going from a typical peaks shape like this to an unresolved cluster of peaks all mixed up together like this. Another important class of MOAH markers are thiophenes, so sulfur ring compounds like benzo, dibenzo, and naphthobenzothiophene that can be easily detected and separated from 3-ring MOAH using, special spectral filters given us by the MES, but are unfortunately strongly reduced by oxidation and refining oil processes. Regarding MOAH collusions, the number one, the most frequently seen, is taken by terpenic olefins. They are typical of natural food metastases and can elute in different fingerprinting and different spaces in the chromatogram. Depending on the number of isoprenic units, they can be small or more complex, like squalene and carotenes. They can be removed at a certain level by epoxidation with some more markers as well, unfortunately. When we are in presence of paper packaging and generally pine resin derivatives, most probably we will detect abietic acid cluster, and so we can easily remove them from quantification. Of course, as for ROCH seen before, the partially and non-hydrogenated fraction of hot melt adhesives is called ROA and elutes in clusters similar to ROCH in the MOAH fraction. When MOSH and MOAH fractions boundaries are not well defined, we most probably see partially polar and heavy naphthenic hydrocarbons in the MOAH fraction, in particular when high olefin concentration samples are present. But how all those data be reported? First, a system verification parameter summary should be written containing all the sample history, concentration, pretreatments applied, because we have seen they can change, and how much sample has been injected per fraction, all the relevant details or references used for classification, drawing, their amount injected to assess detection capability of the instrumentation. Then all chromatographic traces, from spectra masses for filters, and GC per GC counterplots should be reported, as well as the description of all the classes that we have found, search yes or no. And finally, tentative identification of the contamination sources can be hypothesized according to the suggestion given us by the guidance. Just a few little words about quantification of MOSH and MOAH in GC by GC FID. Well, when comparing all to new plot, we must take in account that due to the advanced separation power of GC by GC, most probably the humps have been turned to series of peaks, and that a different separation mechanism, reverse configuration polar versus totally non-polar, has been applied. So the match between the two plots can fit at 100%. Next, we must deal with the limited classification because we miss spectral filters from the MS, because we have all FID detection, and we'll probably need to report some macro classes than singular classes, and not all pollutions can be quantified alone. It's not easy to draw traditional C fractions like, the traditional one, and perhaps only C10 to C50 total quantification should be reported. When we are in presence of interferences, overlapping interferences, we might be aware that cutting away all of these interferences, all these spaces from the chromatogram can lead to an underestimation of what's below, while keeping too much, of course, can lead to an overestimation of the data. And most probably, the correct result is between these two cases. For the same reason, also a powerful tool like the smoking factor algorithm for trimming riding peaks should be applied in a reasonable way. Lack of standardized methods creates, of course, lack of harmonization between laboratories for parameters like data reporting, LOQs, references used for quantification and classification. And of course, to fix and harmonize all these issues, a guidance for quantitation too is needed. Just to summarize what we have seen until now regarding qualitative analysis, we have seen that it should be mandatory every time we don't have compliance samples or maximum level or indicative level are exceeded. Is recommended every time we need an inspection and further investigation on mineral oil contamination sources in the whole food supply chain. Is the only technique that can give us a complete fingerprinting of mineral oil contamination with particular attention to MOAH subclasses and risk assessment. And of course, thanks to the European guidance, now has been well harmonized, and also data reported and interpretation are quite fine. When we go to the quantitative part, we suffer from a lack of harmonization, and more robust and normalized methods are needed to set all the system parameters that I've tried to explain to you before. So traditional SC GC FID analysis is very fast, reproducible, and optimum for routine screening, but is not selective. And since interferences can be superior and detected, we tend to have false positive results, and sometimes interpretation of chromatogram cannot be easy. On the other way, new GC by GC of FID analysis technique is the optimum for mineral oil confirmation. Yes, of course, thanks to the high selectivity and specificity and improved separation power. Is an important tool for risk assessment and evaluation of MOAH by number of rings. For the quantitative part, we have seen that the guidance for laboratory harmonization is still needed. And since now this technique needs more complex and expensive equipment and more time of analysis and, of course, specialized expertise. Okay, this is the end of my presentation. I hope you had a good time. I'm now stop sharing my screen. Yes. Thank you for joining me.
Thank you, Erika.
Thank you, Erika. Thank you, Barbara and Cristiana, for your interesting scientific presentation. So we are to the Q&A question. We try to summarize the more frequent questions we received during the registration. And so we start with the first one. So, are there any raw materials, foodstuff that present particular challenges for MOSH and MOAH analysis? And how high beta-carotenes present in the sample can affect the final results? So, Cristiana, do you want to try to give us a point of view?
Thanks for your question. Some raw materials and foodstuff can present challenges for MOSH and MOAH analysis because of matrix interferences. In particular, high-fat foods and products with a high content of natural terpenes or essential oil, for example, spices, carotenoid-based products, supplements, may complicate chromatographic separation and data interpretation. As seen before, these matrices often require dedicated cleanup steps to avoid interferences in both MOSH and MOAH fraction. In particular, carotenoids are highly hydrophobic compounds that may elute in MOAH fraction while using GC GC FID analysis. So, if not removed during sample preparation, they can contribute to the unresolved signal and lead to an overestimation of MOAH content. For this reason, specific cleanup strategies are typically applied to minimize this kind of interferences and ensure reliable results.
Thank you, Cristiana. So the next question: what impact does the packaging of the samples have? So interaction between packaging and samples. Barbara,
Yes.
talking about food contact materials.
Yes. The packaging may be a source of mineral oil contamination. It is important to know that the mineral oil concern started in the food packaged in a recycled paper and Utah bag. And this kind of packaging may be a source, but the polyolefins may introduce POSH, polyoligomeric saturated hydrocarbons, that may cause an overestimation in MOSH results. It's very important the use of the packaging in sampling, for example, to reduce or minimize this kind of contamination.
Thank you. So, third question. What do you do if you suspect interferences in the chromatograms? Okay, Cristiana or Barbara?
Cristiana, is
Barbara.
okay if I do?
Yes.
Okay.
Yes.
And as showed in the previous slides, when the shape of the chromatogram is not a AMP, the presence of interferences is suspected. And in case of some specific molecules like POSH or PAO, polyalpha olefins, the chromatographic shape is atypical. And on the basis to the shape is possible to hypothesize the interferences.
Okay, thanks again. And is it possible to analyze mixed tocopherol matrix and herbal extract? Okay. Maybe this could involve Erica and GC by GC.
Yeah, I know that there is complex and challenging matrices, and yeah, for herbal and spices matrices, yes, of course, it's possible, but we must take in account a step-by-step approach in order to separate MOSH and MOAH efficiently, avoiding interferences. When we talk about tocopherols mix, we should have more issues, unfortunately, because tocopherols tend to collude in both fractions. So the MOAH one in particular will be highly interfered by tocopherols and tocotrienols. Due to the high amounts involved, purification steps will be, I think, purely applied, and LOQs will be erased. In all of these cases, the search for oil contamination will be better applied to singular raw materials instead of the final product, also taking in account the different limits on food and raw materials.
Thank you, and I exploit that you are okay. And the five, because we receive another question, but we will return on GC. But now the fifth question is, which will be the limits on food and how will they affect the limit on raw materials? So it is related to the first presentation. So Barbara, please.
The limits that will be fixed are 0.5, 1 and 2 milligram per kilo for MOAH on the basis to the fatty content, and with the expectation that will be reported in the annex. Okay. For the raw material like oils, the limits are fixed in the annex, and for others not directly reported in the annex, it is necessary to evaluate case by case the percentage in the final food and the factor of transformation process.
Thank you, Barbara. I have another question that I would to give to Erika, because from the chat box we received this question. Have you used a FID detector or a MS detector for quantification? I think it is related to GC by GC to clarify quantification and qualification.
Yeah, we use MS detector for qualification and FID for quantification. Of course, MS detector can do also quantification, but since the mixture of compounds that we are talking about, so this analysis is quite similar to a more untarget one than a target. Yeah, should not be possible to find a reference, a standard peak for every class of compounds. So FID detection is the only way possible if you wanna do detection of all the classes together in one run.
So many thanks. So we have come to the conclusion of our program. We hope you find this webinar useful. We receive other questions and we will try to give you an answer in the next day. First of all, a big thank you to our speakers sharing their expertise. So thank you also to Dana Sanchez and all Cotecna team that help us in the organization of this event. And last but not least, thank you to you all for your participation to join us today. So in the next day, as I mentioned, we will send you an email with containing a survey form and the recording of this webinar and the relative presentation. And feel free to contact us for other questions or requests. You can find our Cotecna and Neotron contact. So thank you again. And have a great day, everyone, and see you in our next webinar. So thank you and bye. Have a good day.
Thank you to everyone.
Thank you. Bye.
Bye-bye.
Bye.